Okay, on to protein synthesis. The body can make millions of different proteins from just 20 amino acids. So let's get to how we get to those amino acids. In the DNA, there are only 4 nucleotides: adenine, thymine, cytosine, and guanine. And so those combinations of those 4 nucleotides is how we're going to get to add different amino acids, those being the building blocks of the protein. So our genetic code is basically a system or a process that enables those 4 nucleotides to code for different amino acids. And then those amino acids are joined in a sequence, and then depending on the sequence of the amino acid, that is how proteins are different from each other. So boiled down, there are 3 nucleotides per amino acid, and then 20 amino acids possible, and all of that differentiation or differences in that sequence is what makes proteins different from each other. So going from the original DNA sequence, when we see a strand of DNA, it's going to be all different pattern of A, C, T, and G. And so we read those letters in a series of triplets. And so one base triplet on the DNA strand is 3 nucleotides. And the reason we read it in that triplet is because those 3 nucleotides are going to code for 1 amino acid. And so when we go through the process of protein synthesis, for every 3 bases on the nucleotide, there's going to be 1 amino acid added to the chain. And when the DNA strand gets read or copied, if you want to say it that way, a strand of messenger RNA is going to be made. And it's going to keep the integrity of the sequence from the DNA. And when we're looking at the messenger RNA strand, those 3 bases that were a triplet on the DNA are now called a codon. It's a little confusing that it seems like everything has 3 names, but on the messenger RNA strand, it's kind of like sending the instructions away from the nucleus, and the sequence is intact. But now we have it on the messenger RNA sequence in a series of codons. There are 64 possible codons. That's just taking 4 nucleotides times 4 times 4, because we're talking about a triplet. And then they are available to represent 20 possible amino acids. So there is definitely some repetition. So there's multiple codons that code for the same amino acid. And there are 3 stop codons. So there's 3 codons that say, okay, we're done, and they don't code for an amino acid at all. There's also 1 start codon, and that signals where on the messenger RNA strand the process of protein synthesis should begin. Okay, I'm going to skip a couple slides and get straight to protein synthesis. Protein synthesis is the process of making a protein. It happens in all of our body cells, except some cells and some immune cells. But all of our body cells contain identical genes. It's just that different genes are activated in different cells. That means different cells are making different proteins, depending on what type of cell they are. So any given cell can use 1 to 2/3 of its genes. Now, I wouldn't say that that's all at the same time, but that's possible. Okay, so the first process of DNA making a protein, and if you look at that second bullet point where it says DNA to an arrow to messenger RNA to a protein, if you can remember that, you've got the whole process down pat. So DNA making mRNA, that's the first step, and that is called transcription. It occurs in the nucleus. I'm going to post a couple of different animations. I think that's the best way that you're going to be able to visualize this more than me talking about it. And then the second step is called translation, and that happens where the messenger RNA is actually coded for the protein. This happens at the ribosome in the cytoplasm, and that's where the amino acids are actually joined together. So there's really only 2 steps: transcription and translation. Transcription is copying the information from the DNA to making messenger RNA. So there's an enzyme that binds to the DNA. It's called RNA polymerase, and it assembles the messenger RNA. So it starts binding these new RNA nucleotides in the correct order. So whatever's on the DNA, it's going to code for or add the opposite base pair for the messenger RNA. So for example, if there's a cytosine on the DNA, the RNA polymerase is going to add the guanine to make the mRNA. The only difference there is if there's an adenine on the DNA, it's going to add a uracil because RNA does not have thymine. Okay, the end of transcription is basically when the RNA polymerase reaches the end of this certain gene that is to be transcribed, and then the DNA helix kind of wraps back up, and so the RNA, mRNA, is then spliced into the correct parts that are needed. And then that mRNA is going to move out of the nucleus and go out into the cytoplasm, getting ready for the step of translation. Again, I'm going to add some, I would go through this notes and watch those animations, but I'll also add some other ones that I like. So the second step of protein synthesis is translation. That mRNA goes out of the nucleus. Remember, the DNA can't leave the nucleus, so that's why the mRNA is so necessary to keep that sequence carried out. And so that goes out to the cytoplasm and it reaches a ribosome. At the ribosome, a transfer RNA brings one amino acid for every codon that it reads. So for every 3 bases on the mRNA, the transfer RNA arrives with the correct amino acid that codes for or matches up with that codon. And again, it's the sequence of these amino acids that makes each protein different. So the mRNA starts where AUG is on the mRNA, and then for every 3 mRNA codons, or for every 3 bases, 1 amino acid is brought by the transfer RNA. This is a very weird looking transfer RNA. Normally they look like a T, but you can see there's at the bottom is the anticodon that matches up with the codon on the messenger RNA. This is going into a little bit more detail than we need, so I'm going to skip a little bit. Again, if you're watching these animations, I think they're going to do a better job, but there's a better looking tRNA, and see how it's bringing that blue circle. That's the amino acid, so I would definitely watch through this video. That's a much better looking tRNA. But at the end of translation, you can see that up where it says protein, that is a sequence of amino acids, or kind of a chain of amino acids, and that is what a protein is. And so after each amino acid is brought by the tRNAs, they are all joined together to make a protein. Those are some weird pictures. I don't know about these pictures, but I'll attach some better animations. These are making it more complicated than it needs to be. Okay, we talked about the cell organelles that have to do with protein making. So ribosome, this is all happening at the ribosome. And then at the end of translation, a protein is done, except that sometimes proteins are sent to the Golgi apparatus to be packaged, some finishing touches, and possibly exported out of the cell. Okay, this picture is perfect. So this is showing you all of the steps from the original DNA, and then in between step 1 and 2, it's just showing you that DNA, only a section of that DNA is going to be coded for, and that is what's going to be copied or essentially sent out of the nucleus. So in between steps 2 and 3, that would be transcription. And then in between step 3 and 4, really step 4 is showing you translation. And then your final step 6 is the protein made during protein synthesis. This is a very good summary of all of the steps.